Hi everyone. In this video I want to talk about meiosis, a little bit about how it works, and why we think there is meiosis. Okay, so in contrast to mitosis, we sort of understand already why mitosis exists. If mitosis didn't exist, I would still be a single fertilized egg. Right? Mitosis is necessary to make a multicellular organism or for a unicellular organism to make more copies of itself. But meiosis, it's not quite as clear. So the main reason that we think that there's meiosis, and the main consequence of meiosis, is that it reshuffles existing variation, allowing selection to eliminate harmful mutations. So a little bit confusingly, we're going to go back and forth between 2 and 3 chromosomes, because they're useful for showing different things. Cells with 2 and 3 chromosomes. So let's start out here. So as before, different colors indicate different homologous chromosomes. So these green chromosomes are the same. That would be like the chromosome 1 from this cell, and this would be the chromosome 1 from this other different cell. And same thing with the oranges, chromosome 2 and chromosome 2. And I've indicated that these cells have slightly different chromosomes by just coloring this one thick, making this one thick for both chromosomes, and for both chromosomes for this cell to be thin, just so we can follow them. And then I've also indicated that with X's, harmful mutations, here one occurring on chromosome 1, and here one occurring on chromosome 2. And so I just want to point out, to motivate that if this cell were only to divide by mitosis, then all descendant cells would carry this harmful mutation. And if this cell were to divide only by mitosis, all descendant cells would carry this harmful mutation. And as we'll show, meiosis sort of reshuffles things up. It's going to make some cells that have both harmful mutations, but also make other cells that have none. So let's go into the structure of this a little bit. So here we're going to talk about fake meiosis, which is we're going to learn meiosis skipping one important step, which is a DNA duplication step. And the reason for it is, though it's important to understanding exactly what happens, I think that duplication really confuses things and makes it hard to follow what's actually going. I think, for me, the logic of going through it without that duplication makes a lot clearer what's going on, and then we'll add the duplication in later. Okay, so remember in our last lesson we learned that that meiosis and fusion are this cycle by which you go from haploids to diploids and back. So here what we're seeing is 2 cells, again colored with different thick and thin lines, fusing to give rise to a diploid cell. And now this diploid cell is going to have to divide to give rise to 2 haploid cells. And so, as we said before, we have this problem. We can't just divide it in half, because we would end up with this cell having 2 blue chromosomes and this one having 2 green. So, as before, we're going to line things up on the metaphase plate, and we're going to divide. We're going to use tension in the microtubules to pull these 2 apart, so that we get blue, orange, and green on one side and blue, orange, and green on the other side. So, what's wrong with this? Does anybody see something that's funny here? So it turns out to be the case. Well, what you notice is that all the thin chromosomes are one side and all the thick chromosomes are on the other side, which would mean basically that we would get back the exact same cells that we started with. Okay, so for 3 chromosomes, that's possible. The chance of this happening, I think, is 1 in 4. But I do want to point out that it doesn't have to be this way. That the thick and the thin for the blue chromosome are going to line up independently to the thick and the thin for the orange, and independently from the thick and the thin from the green. So we could have all kinds of combinations here. We could have this one cell end up with all thicks, or with a blue thick and a green thick, or with just a blue thick, or with just an orange thick. Each pair is going to line up independently, and that's crucial for this reshuffling effect of meiosis. So if we think about what happens then when these divide, then now we have two new cells, each of which contains a proper set of chromosomes, but they're different, right? This one is a different combination that we haven't seen together. Instead of being thin, 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 or thick, 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 it's a combination. Okay, so let's think about what this means for harmful mutations. So here we have, again, our two cells, each carrying a harmful mutation on a different chromosome. So they're going to undergo fusion to make a diploid. And then they're going to undergo meiosis. And as we said, we can get any of these combinations. So let's think through it. So how many harmful mutations does this top cell carry? Go ahead and pause if you want to think about it. Okay, so actually this cell looks just like this cell. It has both thick chromosomes. This is the case where we've gotten back our original cell genotype. And so in this case we would have one mutation, right? Just the one on the orange. Similarly on this cell looks just like this one, so we would get the green mutation, but not the orange mutation. So this one would have one. So no changes there. But then let's look at these two. So what about this cell? Go ahead and pause if you'd like a minute. So notice that this cell carries the thick orange chromosome, so that's going to have this harmful mutation. And it carries the thin green mutation, so, the thin green chromosome, so that's going to carry this second harmful mutation. So actually this cell has the worst of both worlds, right? It has both harmful mutations. But then what about this cell? Well, here this is the opposite. So you see that it does not have a thick orange chromosome. It has the thin orange chromosome without the harmful mutation. And it has the thick green one without the harmful mutation. So now from our two cells carrying harmful mutations, we've created a subset of cells that carry no harmful mutations. And so what we've done basically is these two cells that start out with problems, they've created a diversity of progeny and a diversity of offspring, and some of those offspring are superior in genotype. They're carrying fewer harmful mutations. So this allows selection to favor these cells and therefore to help eliminate mutations. So to summarize, before meiosis, 100% of cells carried one harmful mutation, but after meiosis, a full quarter of them carry 0. So this is going to allow selection to act. Okay, well, now I'm going to make a second argument. So again, we're going to follow two mutations, but I'm just going to point out that it allows for different helpful combinations of mutations. So now we're not going to talk about harmful ones, but let's imagine that mutation 1 or mutation 2 by themselves, they're okay, they're fine. But that mutation 1 and 2 together are going to increase fitness of a cell. Okay, so now let's trace out what we expect to see. So which of these cells has the combination of 1 plus 2? Go ahead and pause if you'd like to take a moment. Okay, so if we go through it, we'll see that D is going to carry both chromosome 1, because that's, mutation 1, because that's on the thick orange chromosome, and mutation 2 because that's on the thin green chromosome. And so now, what this means is that if these cells were only to have divided by mitosis, then all descendants would either have 1 or 2, but not both. But meiosis has allowed us to create a combination that carries these two mutations that work well together. These are basically the two reasons that we think that meiosis is important and why it's seen so much across eukaryotes, is that it's important to get this reshuffling of variation. Notice this is not like mutation, right? It's not a new mutation being created. It's taking the existing variation, that is mutation 1 versus the non-mutant, mutation 2 versus the non-mutant, and it's reshuffling them and making new combinations. Okay, so now let's talk a little bit about the products of meiosis. And so we can remember that with some organisms like yeast that stressful conditions tend to lead to meiosis. So during non-stressful conditions, that it'll mostly be mitotic division, but that stress can bring on meiosis. So if we think about this, so when things are going pretty well, when the organism seems to be dividing without problems, when the organism is not stressed out, then just keep dividing to give rise, the organism's going to keep dividing to give rise to more. But when some challenge has arisen, when there's some stress, when the environment is posing a challenge, then it might be a good strategy to try to create a diversity of cells, a diversity of offspring, in hopes that some of these offspring may have a genotype, a combination of genes, that is fit in this environment. So under these circumstances, it makes sense to do this recombination to create a diversity of progeny. So this all fits with our notion of that meiosis might be important for creating, that the reason there's meiosis might be because it optimizes the fitness of all the descendants together by creating some which are very fit. And so I just wanted to point out the difference between the fake meiosis we're discussing and the real meiosis is that there's this round of DNA duplication. We'll return to this in maybe in next lecture. So I do want to point out here that this meiosis, this fake meiosis that we're looking at, it looks a lot like mitosis, right? Basically, when we showed mitosis, we used this exact same slide, except for in that case, there wasn't this difference between thin and thick. This is confusing because it makes it hard to remember that in meiosis, what's lining up here is different than what's lining up in mitosis. Okay, so in mitosis, the two blue chromosomes are actually exact duplicates of each other. Remember, the way that we end up with those two chromosomes in mitosis is that the DNA has just duplicated. In this case, it's different, right? The relationship between this blue chromosome and that blue chromosome are that they arose by fusion of a cell that contained this chromosome and a cell that contained this chromosome. If those cells aren't very closely related, that means that these two chromosomes are going to be slightly different. Right? So here we're looking at differences in alleles of these two chromosomes instead of identical chromosomes. Okay, so that's what I'm pointing out here. These two chromosomes that line up in meiosis are not identical copies. In mitosis, they are identical copies.